Welcome to Quantum Frontiers. Today, we're tackling one of the most intriguing and debated topics in quantum biology. Can quantum phenomena drive the self-alignment and orientation of molecular properties and actions during chemical reactions, leading the biological cell self-assembly?
Indeed, this question addresses the heart of how molecular systems organize and communicate. While classical biology explains molecular interactions well at short distances, phenomena like long-range coordination or the synchronized alignment of molecules hint at a more subtle mechanism. Could quantum mechanics provide that explanation? Or is the challenge simply too great to tackle experimentally?
Let's start with long-range signal transfer. From a quantum perspective, phenomena like entanglement or coherence could theoretically mediate signals over significant distances. However, living systems are warm, noisy, and chaotic, conditions that typically destroy quantum coherence almost instantly. This makes experimental proof extraordinarily difficult.
And from a biological perspective, the complexity of cellular systems compounds the challenge. Molecules and cells don't exist in isolation. They interact constantly with their environment, creating a dynamic network of reactions. Untangling these interactions to isolate a quantum effect is like trying to listen to a single instrument in a symphony while the orchestra plays at full volume.
And a good analogy, even if coherence or entanglement exists briefly, measuring it in such environments is nearly impossible with current tools. Quantum states are incredibly delicate, and their signatures are subtle. Experimental setups require extreme isolation, often at cryogenic temperatures, which is incompatible with studying living systems.
That's where the biology community has concerns. Cryogenic or simplified conditions don't reflect the true environment of living cells. Molecules in a living cell are subject to thermal noise, ionic fluctuations, and constant collisions. For quantum effects to play a meaningful role in self-alignment or signal transfer, they'd need to persist under these conditions, which seems highly unlikely.
Yet there are examples that challenge this assumption. In photosynthesis, for instance, quantum coherence appears to guide energy transfer efficiently through disordered molecular networks. If similar coherence mechanisms exist in other biological systems, it could explain how molecules align their motions or coordinate reactions over long distances. Why is it so hard to prove this?
Part of the difficulty lies in distinguishing quantum effects from classical processes. For example, in enzymatic reactions, enhanced efficiency might be attributed to quantum tunneling, but classical models like induced fit or substrate dynamics could also explain the observations. Without direct evidence, attributing molecular alignment or long-range coordination to quantum mechanics risks overinterpretation.
Another issue is the timescale. Quantum effects like coherence or tunneling often occur on femtosecond or even attosecond timescales. Capturing these fleeting events requires ultrafast spectroscopy and precision that's difficult to achieve in complex, dynamic systems like cells.
And even if you can capture them, there's the question of causation versus correlation. Just because you detect quantum behavior doesn't mean it's driving molecular alignment or long-range signals. For instance, enhanced diffusion in enzymes might be a thermal effect rather than quantum-driven self-alignment. How do you isolate one mechanism from the other?
That's a central challenge. Most experiments rely on indirect measurements, like observing reaction rates or molecular dynamics. To conclusively prove quantum involvement, we'd need to manipulate quantum states directly and observe their effects on the system. This requires not only advanced tools, but also precise control over molecular environments, which we're far from achieving.
Biology adds another layer of complexity. In living systems, molecular alignment often depends on networks of reactions and signaling pathways. These processes are inherently stochastic, making it hard to pin down a deterministic quantum mechanism. Could quantum mechanics even operate reliably in such an environment?
Theoretically, yes, but the lack of reproducibility in experiments is a major obstacle. Biological systems vary from one cell to another, and even minor differences in experimental conditions can mask or mimic quantum effects. Reproducibility is critical for proving quantum involvement, but it's nearly impossible in such noisy, variable systems.
That's why the field of quantum biology remains so divided. On one hand, the theoretical framework suggests that quantum mechanics could contribute to molecular alignment or long-range signaling. On the other hand, the experimental challenges and lack of direct evidence make these ideas speculative at best.
Still, the potential implications are enormous. If we could prove that quantum mechanics governs these processes, it would transform our understanding of life and open up new avenues for medicine and technology. Quantum-inspired drugs, precision diagnostics, or even bioengineered quantum systems could become a reality.
I agree, but we must balance ambition with caution. Overhyping the role of quantum mechanics in biology risks discrediting the field if the evidence doesn't hold up. The focus should be on developing better tools and experimental methods to test these ideas rigorously.
Well said. This is an exciting frontier, but one fraught with challenges. The intersection of quantum mechanics and biology could redefine our understanding of molecular systems, but the road ahead is long and uncertain.
And that's what makes it so compelling.
Let's pick up where we left off. One area that deserves more attention is reaction-driven alignment in cellular systems. Some researchers suggest that molecules might align their dynamics during chemical reactions to optimize efficiency. Could quantum mechanics play a role in aligning these molecular motions?
That's a fascinating possibility. Reaction-driven alignment could explain why some biochemical pathways are so efficient, even in noisy environments. For instance, during ATP synthesis in mitochondria, the rotation of the ATP synthase complex is highly coordinated. While classical physics explains much of this, quantum mechanics might provide an additional layer of precision. What's your perspective on this?
It's theoretically plausible. Quantum effects such as vibrational coherence or tunneling could synchronize molecular motions at critical moments in the reaction cycle. However, proving this experimentally is challenging. To isolate quantum contributions, you'd need to control every variable: temperature, molecular crowding, ionic conditions. And that's nearly impossible in a living cell.
Exactly. Living systems are inherently messy. For example, during protein-protein interactions, molecular crowding and thermal noise dominates. Yet these interactions often occur with remarkable specificity and speed. Could quantum mechanics provide a framework for understanding how molecules find each other so effectively?
That's a great question. Quantum tunneling, or wave-like behavior, could allow molecules to sample potential binding sites more efficiently than classical models predict. However, this idea faces resistance because it's hard to distinguish quantum effects from purely probabilistic classical mechanisms. Are there biological systems where you think this alignment is particularly evident?
One intriguing example is ion channel dynamics. In neurons, ion channels open and close with precise timing to propagate electrical signals. Some researchers propose that quantum effects like tunneling might influence the movement of ions across these channels. While it's a speculative idea, it could explain the extraordinary speed and precision of neural signaling.
Ion channels are a good candidate for studying quantum mechanics because they involve small ions and narrow pathways, conditions where quantum effects are more likely. But again, the question is whether these effects persist long enough to influence biological function. What about larger scale behaviors like collective cellular migration during wound healing or tissue formation?
Collective behaviors are fascinating. Cells coordinate their movements over long distances, often aligning their motion to achieve a common goal, like closing a wound. While classical signaling pathways explain much of this, the precision of alignment raises questions. Could quantum coherence or entanglement be at play here, perhaps indirectly influencing cellular coordination?
Possibly, but proving it would be an uphill battle. Collective behaviors involve so many interacting variables that isolating quantum contributions is nearly impossible. Instead, researchers might focus on smaller, simpler systems, like molecular motors or enzymes, to build up evidence for quantum effects.
That's a practical approach. Speaking of simpler systems, let's discuss enhanced enzymatic diffusion during catalysis. Some experiments show that enzymes move more actively when catalyzing reactions, as if they're self-propelled. Could this phenomenon be quantum-driven?
It's an intriguing idea. Enhanced diffusion might arise from reaction-driven forces, but quantum mechanics could amplify these effects. For example, quantum tunneling could reduce the energy barrier for catalysis, creating localized energy changes that drive motion. However, thermal effects or conformational changes could also explain the observations, making it hard to pin down a quantum origin.
That's the recurring theme, isn't it? For every quantum hypothesis, there's often a plausible classical explanation. Let's shift to molecular signaling within cells. Signal transduction pathways rely on rapid and specific communication between molecules. Could quantum entanglement play a role here, allowing signals to propagate more efficiently?
Entanglement is a tantalizing idea. If molecules were entangled, changes in one molecule could instantaneously affect another, even over long distances. However, decoherence would likely destroy entanglement before it could influence cellular signaling. Unless biological systems have evolved ways to protect entanglement, like error correction mechanisms in quantum computing, this remains speculative.
Biological systems are resourceful, so it's not entirely out of the question. For instance, protein complexes might create microenvironments that shield quantum states temporarily. Could such mechanisms explain phenomena like proton-coupled electron transfer in respiration?
Absolutely. Proton coupled electron transfer involves both quantum tunneling and long range coordination, making it an ideal candidate for quantum biological studies. Understanding how these reactions occur so efficiently in noisy environments could unlock new insights into how biology utilizes quantum mechanics. Star Trek.
Agreed. This brings us to quantum-installed medical technologies. If we understand how biological systems manage quantum effects, we could design quantum-installed drugs or diagnostic tools. For example, quantum sensors might detect subtle molecular changes that precede disease.
That's an exciting application. Quantum sensors are already being developed for use in imaging and diagnostics, but integrating them with biological systems is the next big challenge. Another possibility is quantum-enhanced drug delivery. By targeting specific quantum states or pathways, we could create treatments with unprecedented precision.
That's a bold vision, but it depends on overcoming the fundamental challenges we've discussed today. Without solid experimental evidence, quantum biology will remain a speculative field. What do you think is the most critical step forward?
Developing tools to observe and manipulate quantum effects in real time within living systems. Advances in ultrafast spectroscopy, quantum-enhanced microscopy, and computational modeling will be essential. But equally important is fostering interdisciplinary collaboration. Physicists and biologists must work together to test these hypotheses rigorously.
Absolutely. Bridging these disciplines is the only way to unravel the quantum mysteries of life. To our listeners, thank you for joining us on this journey through the cutting edge of quantum biology. Stay curious, stay critical, and keep exploring the unknown. Until next time on Quantum Biology Frontiers.